Welcome to our Train the Trainers webinar of the National Center for Health Research. I'm Dr. Diana Zuckerman. This webinar will give you the information you need to train patient advocates to understand the basics of the kinds of clinical trials that are needed to prove that a medical product is safe and is effective. Our workshops include a lot of opportunities for participants to share their views, ask questions, and network with other patient partners. We won't be showing that in this webinar because we'll be focusing on the content of our workshops, not on the process. We're assuming you know the importance of an interactive learning experience and the value of giving patient partners time to tell their stories and get to know each other. So, to keep this webinar under an hour, the focus is on the information that engaged our patient partners and energized them to be national advocates, sharing their perspectives about what they need to know about a treatment's risks and benefits for them. We will start with a shortened version of one of our keynote speakers talking about her journey as a patient advocate. She spoke after we had gone around the room so that each participant had already introduced themselves. We found that no matter which patient advocate we invited to give the keynote, it was one of the most highly rated sessions of the workshop.
How many of you actually knew what it was or learned about being a patient advocate when you were in middle school, high school, college? Because I think patient advocates, it's a wonderful role, but it's not something that has always been talked about. It's something that, unless you've been touched personally, you don't really think about it in a great way. People just go through life. And for me, as I mentioned earlier, and I've listened to many of you around the table earlier today, you spoke about what brought you here. And I think that it's so important for us to realize being an advocate is really learning on the spot. I like to say it's drinking out of a fire hose because you had no idea, you had no preparation. You just found yourself in a position and you said, no, I'm not going to take it sitting down. I need to find a voice and I need to do something. And so we basically are self-taught for most of us because at the end of the day, there's really, I actually did look. There is no degree, bachelor, master's, or PhD to be a patient advocate. And so you're basically trying to figure it out. And you have an opportunity of coming here, and I'm sure many of you have gone to other events where there are conferences and advocates come together. And that's how you actually learn more about what you're trying to do and how to accomplish it. But I will say to you that being a patient advocate can be a frustrating journey because you have no book. There's no fine print. There's nothing that you can read to teach you how to do what you want to do. You want to be able to have a seat at the table for the decision makers. If you're not at that table, all your efforts don't really line up for much. But the question is, how do you get a seat at the table? You can have passion, but that's not going to get you a seat at the table. And when you get to the table, what are you going to do at the table? Do you have evidence-based information so that when you pose, and some people say argument kind of is a little strong as a word, but when you have an argument that you want to make, if you have the evidence to support it, you might have a greater opportunity to get people to listen. So we each have the job to make sure that we go out and we seek the information so that we have the evidence to support what we're saying, because otherwise people are going to say, well, he or she, they just like to make a lot of noise, but nothing they say really makes sense. And you really want to be able to get people to hear you, to listen to what you're saying. I had an opportunity to start doing work with PCORI, where I actually am a consumer reviewer, and I review research material and critique it. And then you have an in-person meeting. And I find that to be really informative because although I'm not a scientist and clearly not going to school to be that, you have the opportunity to sit around a table. And there you have clinicians, you have scientists, you have other patient advocates, and it's a group discussion about these respective proposals. And it is so enlightening to, because when I go there, I'm coming with my patient advocate hat, and I'm concerned about how is this going to impact someone's quality of life? Because I think, when you've been impacted by disease or anything else, illness, quality of life is really key to you. That's really what's important. But oftentimes, at least years ago, when research was done, it was about the next drug. And it didn't matter whether the drug made you feel good. It was like if it kept you living a day longer, they thought that was a success. But as a patient, that's not what we believe. And so having the opportunity to bring the patient voice is really important. But also having the opportunity for a scientist to explain the science of it all, and whether this study actually can work based on how it's designed, I think is really helpful in terms of education as well. And so for me, I would just encourage all of you, for whatever your respective cause is to really figure out who you can connect with in order to be able to broaden your knowledge. I know the Kims, said they had no clue as to what they were doing, and you still don't know whether you know what you're doing, but you have numbers now. Okay, you've been able to get supporters, and you know that there's interest out there. And so it's really important for you to be able to connect. I like to say you need to try and form, whether it's a small army or a large army, but an army nonetheless, of like-minded people who are looking to push further. Because when you may have that day where you're not as energized, there's someone else in the group that can continue pushing forward, and you can take turns, because when you try to take advocacy on by yourself and you get weary and there's no one else around, then things just fall to the wayside, and all the momentum that you've gathered, it then diminishes. As an advocate, you definitely focus on you have to drive this car, and you have to build this relationship, and you have to actually make demands of this relationship. And if you don't let people know that you are looking to be a partner, then someone is going to start to make decisions for you. And that's where you lose your voice. And so just encourage you, however you can find your voice, please take the time and figure out what works best for you. If you're an introvert, an extrovert, we all don't like certain things, but know what you're good at and run with that, because that's when you'll be the most effective, when you're doing something that you love, that you're passionate about, and you're capable of, because you don't want to do anything that's going to stress you out. And there's no need to, because you could have a partner in your army that they can pick up on an area that is not your strength. But together, advocacy can go a very long way if we all continue determining what is the best road for us, and in partnership with other individuals as well.
The next part is an edited version of my talk, which set the stage for what we were teaching. Patients want treatments that are safe and effective, but they don't necessarily know how decisions are made about what treatments are considered safe and effective. And they don't necessarily know whether that means a treatment will work for them. We explain how the FDA makes decisions about what drugs and medical devices can be sold in the United States and what the implications are for each of us. There are always a lot of questions during this talk, but we have edited them out to keep it shorter. Safe and effective means the benefits outweigh the risks for most patients. That's probably not how most of us think about safe and effective, but that is the definition for FDA. So keep in mind that it's okay if most patients don't benefit from a drug. So for example, many cancer drugs don't work on most patients. They work on maybe a third of the patients or something like that. But as long as they're not terribly harmful to most patients, then that risk-benefit ratio will still be considered reasonable. And also to be sure that you understand that the FDA by law cannot consider cost. So the FDA knows that when a new drug is approved, it's probably going to cost more than the other drugs on the market, and it's especially going to cost more than generic drugs. But that FDA cannot consider cost, so they do in fact approve a new drug knowing it might not be as good as an older drug that costs 10% as much or something. These are the things that it doesn't mean. FDA approval of drugs or devices. It does not ever mean that nobody will die from this product. It never means that. It doesn't ever mean that few people will be harmed by this product. It could be that people will be harmed by the product. Again, it's a ratio of benefit to risk. It doesn't mean that the product is safe for long-term use. Almost all the time, FDA requires studies that are relatively short-term. Even if the study might be 2 years, it might be for a cholesterol-lowering medication or a pain medication that might be used for many years. So it's pretty much never true that a product is going to be studied for as long a period of time as people are going to end up taking it. And as I just alluded to, FDA approval never means that this is proven more effective than any other product on the market or cheaper products on the market. Sometimes they are more effective, but that's not the standard that FDA has. By law, that is not the standard. Clinical trials are important. Clinical trials are studies of humans, which are used to prove, in the case that we're talking about, used to prove whether product is safe and effective. And as we think about what are the outcomes that matter to you, and I'm happy to add a few to this list, and obviously it depends on what the illness is. So it might be survival, especially for something like cancer or heart disease. It could mean fewer days in the hospital for the next year or maybe several years. It could mean fewer serious or unpleasant side effects. So a side effect can be very serious, can be deadly. But if you're nauseous all the time, that's not serious, but it's so unpleasant that it just might be unacceptable anyway. And then other symptoms that can result from devices or from drugs, such as pain, such as nausea. The main focus for drug approval is that it's safe, but that's safe but usually based on 2 short-term clinical trials, and that it's effective, usually, although not always, compared to placebo, or in the case where it would be unethical to compare it to placebo. So for example, for cancer drugs, you might have the new cancer drug plus the old cancer drug compared to the old cancer drug plus placebo. So nobody's getting nothing, but you're still comparing it to placebo. It's just a combination. And another way that that the studies can be done faster is to look at surrogate endpoints. And Helen mentioned this, and I think all of you have at least heard the term before from the intro workshop or from other reading. So biomarkers or surrogate endpoints are things that you measure that aren't really your health. So we think of lower cholesterol as being our health, but actually it's more of a what might be called a marker or a surrogate for health. So same with glucose levels, same with bone mineral density, same with progression-free survival in cancer. I'll give you some examples of this. So a drug can lower glucose, sugar levels, but still not help diabetics live longer or healthier. That has happened. Or you can have a drug that's really good at lowering blood pressure, but somehow the patient's lives are not being saved. Or you can have a screening test to prevent death from cancer, but the patient won't necessarily live longer. And that's, for example, x-rays for lung cancer is a good example of that, that people get x-rays, used to get x-rays for lung cancer, and they could get a detection of lung cancer earlier, but by the time it was large enough to show up on that x-ray, it was too late to do a successful treatment or surgery. Similarly, chemotherapy can kill cancer cells, and if the surrogate endpoint is shrinking a tumor, then it looks like it's working, but it doesn't necessarily help the person live longer, and it can make their life miserable. So the key question for all of you as patient advocates, as you think about research, is do you want biomarkers or surrogate endpoints to be used in research? If they are used, what kind of protections do you want? What kind of safeguards do you want to make sure that those surrogates really will predict a healthy outcome, and making sure that research is done about healthy outcomes, not just about these surrogates. So again, I'll just go to the example of cancer, which is of concern to many of us, even if we haven't had it ourselves, because who doesn't have a loved one or a friend who's had cancer. 2/3 of all cancer drugs in recent years are now approved on the basis of surrogate endpoints, and that surrogate endpoint is usually going to be shrinking a tumor. And then the FDA will require what they call a post-market study. All a post-market study means is it's a study done after it's already on the market. In other words, it's already approved, it's already being sold, and now it's being studied some more. That's what a post-market study means. For the FDA, they'll say, we're going to approve this cancer drug on the basis of the fact that it shrinks tumors, but now you have to do a longer-term study. It will take longer to do, usually a few years longer, to see, so you can shrink a tumor in 5 months or 3 months, but you might have to study it for a couple of years to find out if people actually live longer. So the FDA will say, you have to prove that your product will help people live longer. And then the FDA will give a few years to the company to do those post-market studies. And what some researchers have found is that most of these cancer drugs, of these 2/3 cancer drugs that were approved on the basis of surrogate endpoints ending in about 2012, so we now have a few more years to have done additional research and to look at it, most of them have not yet, and perhaps never will be, proven to prolong life. And our center has done some research also showing that they're not necessarily improving quality of life either. If you look at the post-market studies, there's no evidence that these cancer drugs are really having a benefit. And talking about post-market studies, what's the difference between pre-market studies and post-market studies? When pre-market studies don't provide evidence of living longer or better, then FDA usually will require a longer-term post-market study in order to gather more information. But think of it this way, the product's already on the market, people are already paying for it. So as a patient, if you're taking the drug, whether you're in a post-market study or not, you're basically paying sometimes a lot of money, such as in the cancer drugs, paying to be a guinea pig in an experiment for a product that isn't proven yet. And the other part of this that I find particularly disturbing is that the company has so little incentive to do that study quickly, to do it well, to include a diverse group of patients. So the FDA might say, well, you don't have enough women in this study, or you don't have enough people over 65, or you don't have enough people of color. So we want to make sure that when you do your post-market study, you have more of those different groups. But there's nothing that requires them to do that. They're only saying, we'd like you to do it, and sometimes they do it, and sometimes they don't. So let's go through the criteria for device approvals, which by law are different than for drugs. So drugs have to be safe and effective, and we talked about what that means, benefits outweighing the risks. Devices have to be reasonably safe and reasonably effective. Again, similar, the benefits should outweigh the risks for most people, but it's a little unclear, but clearly seems to be saying the standards are lower, reasonably safe, reasonably effective. Let's talk about what that means. More than 95% of medical devices are not required to be studied in clinical trials. The FDA does not require clinical trials at all for over 95%. And that includes hip replacements, knee replacements, shoulder replacements, most of those joints. Actually, a fair number of cardiac implants don't have to be studied in clinical trials. Many, a really kind of shocking number of devices don't have to be studied. Let's look at that visually. The lowest risk devices are not tested at all in any way, shape, or form. And those include things that you might say, okay, fine, Band-Aids, tongue depressors, crutches. I like the example of crutches because, that's a low risk product unless it doesn't work. If you've got crutches that break when you try to walk with them and you fall down, you can be really hurt by that, permanently hurt by that. So even a low risk device has some risks, but the feeling is any reasonable company will know how to make these devices, and FDA does not ask them to submit anything about their product. The moderate risk devices, which is a huge percentage of devices and includes a lot of different things. So here you have a hip replacement graphic in the middle. You have the inferior vena cava filter, which is a filter that's implanted in the body to filter out blood clots. That seems very high risk, but they call that moderate risk because it's supposed to be temporary. You have a glucose meter to measure blood sugar. Again, that might seem moderate risk unless it's inaccurate, in which case it could be fatal or very, very harmful if it's so inaccurate. And then contact lenses and contact lens solution. Contact lenses are devices, and contact lens solution are also considered devices. These are moderate risk devices. None of these products studied in clinical trials unless the company wants to. The FDA doesn't require it. And about 98% of devices that are on the market with some kind of regulatory activity by the FDA, some kind of FDA review, are these moderate risk devices. They're reviewed through something called the 510K process. That just refers to a section of the law that happens to be numbered 510K. They're not tested for safety or effectiveness, but instead these products must be substantially equivalent to some other devices that are legally on the market. So there are no clinical trials required except under very rare circumstances. No inspections of the manufacturing facility to make sure it's being made the way they're supposed to be made. And prior to selling it, they might have inspections later if there's a problem, but not before they're sold. And again, by law, the FDA cannot require post-market studies when they first clear these. It's not called approval. When they first say, yes, you can sell these hip replacements, it would be logical if there's no clinical trials to say, yes, you can sell them, but we'd like to see clinical trials. By law, they can't do that. So that's part of the beauty of this process, is that they have to be substantially equivalent to another device that's already on the market, which itself was never had to be proven safe or effective in clinical trials, because it's a little bit complicated, but the law was started in 1976, and these the new products were supposed to be substantially equivalent to something that was already on the market before 1976. It's called a predicate device in this case. And the ones before 1976 had never been tested at all because the FDA had no jurisdiction over devices before 1976. I think the original intent was that as more and more devices changed and got more sophisticated, those earlier devices would no longer be sold, but instead they've just had a train, if you will, of devices. So, let's say you had an implant that was on the market in 1976, and then in 1980 the company changed it, but they said it was substantially equivalent. And then 5 years later, they changed it again, but now it's substantially equivalent to the one that was cleared 5 years earlier, and it just keeps going. And I'll show you some pictures that will illustrate that. Actually, that's the best way to show it. Yeah, Cindy.
Thank you. I just want to say that the clinicaltrials.gov is an excellent resource, and I found my Reveal, it was a Medtronic Reveal XT implantable loop recorder placed into my chest. And since 2008, when the device came out, and it's now 2016, I'm looking at it right now on my computer, no study results posted. That's 8 years. So if you have something that you want to look up and see if there's any studies, that's a great resource to use to find that out.
Well, let me say that unfortunately the fact that it says that doesn't mean that there is no studies. It just means that they haven't been submitted to clinicaltrials.gov. I mean, it might well mean that there's no studies showing that this is a good product, because of course when it's a good product, then the company wants everybody to know about it. But you can't, I mean, that's one of the problems with clinicaltrials.gov. You can't assume just because nothing's there that nothing's been done. It might mean that, or it might not mean that.
I've done further research. I haven't done anything yet, but
well,
I'm
as
sure
I said, it could mean that, but you can't assume it for everything. So let's talk about those 2% of medical devices that are considered high risk by the FDA. The middle one is a pacemaker. You might be surprised to know that this silver shiny thing is an artificial heart. And this other thing is an infusion pump, something that would be used in many, many hospitals. These are clearly life-saving devices, but of course they're not the only life-saving devices, but these are clearly very high-risk life-saving devices. They go through a process called the pre-market approval process, or the PMA. The women from Essure are probably surprised. It's a little weird that the Essure birth control device was also considered the highest risk. Why? I think the answer to that is probably more history than anything else, that because IUDs and were found to be very dangerous after women had them for a long time, that reproductive devices ended up with more scrutiny. As we'll talk about later, not very good scrutiny though. So highest risk devices tend to be implants, although many implants are not high risk. They tend to be life-saving, although many life-saving devices are not considered high risk, or life-sustaining. Again, many are not high risk, but some of them are. And they go through a process that's more like the drug process of clinical trials. They must be proven reasonably safe and reasonably effective. Remember, those other devices didn't actually have to be proven to be safe or effective. They only had to be substantially equivalent to a device that was assumed to be safe and effective because it's on the market, but was not proven safe or effective either. So what's different though about devices is, remember I said that traditionally drugs are studied in 2 clinical trials. For devices, it only is 1, and although many drugs are only studied in 1, at least some are studied in 2. All of these device approvals are based on basically 1 clinical trial, almost never double blind. In fact, often not single blind. And almost always a smaller sample. I mean, you have sometimes thousands of patients studied in a drug study, and you might have 40 patients in a device study or 100 patients. It's not unusual at all. So even for these very high-risk devices, they're not being studied on very many people. So a typical device clinical trial might be a controlled but not a random clinical trial. That would be one where the doctors or the patients choose who gets which device, and where they compare the patients receiving the new device with patients who don't. Especially when you have an implant, you can imagine it would be questionable to have surgery and not put anything in. So, there are reasons why double-blind studies are very uncommon for devices, especially for implants. So instead of having it randomized, you might have two patient groups that are similar or that are matched, in terms of age or sex or diagnosis or hopefully all of those things. So that would be a more typical device trial. Not as good as the gold standard of a double-blind randomized clinical trial. But it's also not unusual to have clinical trials with no control group. And Essure is such a good example of that, but not the only example of that. So that's the Essure device in the photo. The patients or the doctors, or both, chose or will choose who gets the new device. And sometimes it's not compared to anything, or it's compared to some kind of general information about women that age in general or people that age in general or something like that. So in a study like this with no control group, all we can tell from the study is how the patients feel. In the case of Essure, did they get pregnant or not? What symptoms did they have? But we don't have anything to compare it to. And that's a real problem. You want to have that control group. You want to be able to compare them to other people who are just like them, but who didn't get that product, in order to say what's the benefit and what's the bad side effects of that product. So then the question might come up, well, is an uncontrolled clinical trial better than no trial at all? And I would say, this is my opinion, if researchers are looking for the truth, then any clinical trial can be helpful, even a badly designed one, if they're looking for the truth. But if the goal is just to prove that their product is safe and effective, and that's really what they want to do, it's a lot easier to do that when you have no control group. And again, I can use the Essure for a good example. You had women coming in saying, I have terrible, terrible pelvic pain. And you had the doctor saying, well, we're not even going to count that because that's not related to the device. How did they know it wasn't related to the device? They just assumed it wasn't related to the device. In some cases, they said, talk to any OBGYN, and any OBGYN will tell you that a lot of women have pelvic pain. The main reason why women go to OBGYNs is because they have pelvic pain. Well, that may or may not be true, but that's still not women in general. So, in my opinion, you always want a control group. And if you had controlled the women, had a control group comparing the women getting Essure with women using some other kind of birth control, whether it was an IUD or anything else, then you would have been able to say they were more likely or less likely to have pelvic pain, they were more likely or less likely to get pregnant, they were more likely or less likely to have any number of other side effects, that it was just easier without a control group to say, oh, that's unrelated. That has nothing to do with it. So, picture's worth 1,000 words. Here's some of the things that FDA thinks are substantially equivalent. These were two types of jaw implant materials. One of them looks like something that you would fix your furniture with, and the other is a sheet of silicone, kind of a mesh. And as you can see, they don't look anything alike. They're made out of completely different material. How anybody could think these are substantially equivalent, I could not say, but FDA did. And by the way, these metal implants did end up doing horrible harm to patients who ended up, in some cases, with holes in their skull and unable to eat anything but liquid nourishment because they could no longer had a jaw that was functional. Here's another device that's a little harder to see, but this was a temporary implant for also for the jaw area. You can see one of them is a small piece. The other one goes all the way around the jaw. One has movable parts, one doesn't. Again, how this could be considered substantially equivalent, it takes a lot of imagination, I think. And here's a spinal system. You might wonder why spinal surgery so rarely works. Here's one device, again, looks like some little screw thing that you would put in your furniture. The other is that metal coming up from the spine, so here. And obviously this has little resemblance to the other. One has moving parts, much more complex. It's changed dramatically since 1996, and yet it's still substantially equivalent and being cleared that way. And the last example is a non-thermal shortwave device for pain. The early one, you can see the person is actually in it. Now it's just put on the limb or whatever is in pain. So again, really different, and yet considered substantially equivalent. By the way, no evidence that either the old one or the new one actually works, but both were cleared. And some of you have seen this photo before. In 1 year that we were studying, almost 500 million devices that had gone through this 510K substantial equivalence review were recalled later by the FDA as being high risk, more than 500 million in 1 year, including the contaminated alcohol swabs that killed this little boy. So, in conclusion, and then I can answer any questions. Remember, the gold standard, 2 double-blind randomized clinical trials that actually study patients' health, whether the health is survival or pain or something that really matters to patients. Unfortunately, today's FDA, partly because of laws that have changed and partly because of pressure, rarely requires 2 double-blind randomized clinical trials that actually study patients' health, and they especially don't require that for fast-track drugs, but sometimes they don't require it for other drugs as well. And they never, ever require 2 double-blind randomized clinical trials for devices, never. More than 95% of medical devices have no clinical trials or proof of safety or efficacy. Just not required. If the company's doing those studies, we don't know about them. And then to get back to the first thing we did today, the ads that you'll see for these products are selling hope, but are not giving you the facts based on the actual studies that have been done. So what are the implications for this? As we talked about before, whether you as a patient advocate care more about the speed of approval of new treatments, or whether you care more about good safety data or good data showing something works. I mean, we talk a lot about safety, and many patients, advocates care a lot about safety, but it's obviously hugely upsetting if you find out that your product is perfect, that your medical product is perfectly safe. It just doesn't work. So whichever you care about more is probably going to be dependent on the options you have. If you're desperate for a treatment that works and nothing has worked, either there's no treatment or you've tried everything already and you're willing to take a risk, then you wish that FDA would move things along faster. But, you know, if you're taking birth control pills and there's 80 different kinds, wouldn't you rather take the ones that are safer? Or if you're, you know, taking heart disease medication, wouldn't you like to take the medication that's safer for you? And wouldn't you like to have good evidence to know what that is? So desperate patients may choose riskier treatments, and they may even be willing to pay for ones that aren't proven. And that is part of the big debate right now about what should the standards be. And that's exactly why FDA is saying we need to talk to patients about this. And we'll talk more today and tomorrow about that. But what's happened is that so many patient advocates are getting information from their doctors and or drug companies about the need for faster approvals, and they're not getting the information about safety. The implications, good quality research does take time. You can speed things up, but if you really want good quality research that's going to prove what's likely to happen, it takes years to do, and it takes time if you're going to replicate it, if you're going to do 2 studies. Now you can do 2 studies at the same time, but companies, you know, very often like to do 1 study at a time because they don't want to waste their money if the first study isn't going to have good results. So that's part of why it takes longer. Meantime, while, you know, if we keep our standards high, if we keep those scientific research standards high, some patients are going to die waiting. They're going to die waiting for a new treatment to be approved. On the other hand, some patients are going to be harmed, the way things are now, by an unproven treatment that has been approved. And that's not even counting the people who are harmed or helped by experimental trials. And again, just to remember that new isn't always better. New medical products may be better or they may be worse. That's why we do studies. We don't know. They may be promising, they may look great. By the time you get to these major clinical trials, there have been some studies that make it seem promising, but there are a lot of drugs and devices that never get through that to the finish line because they start out looking very promising, but somewhere along the line you figure out that they are not nearly as good as we thought. So that's why we want controlled clinical trials, and that's why we want all the groups to be studied, men and women, people of color, people of different ages. We'll hear more about that later, but just to say it takes time to do that. And if we speed things up, we're going to have less of that kind of information.
The next section is a brief explanation of the kinds of clinical trials that are used to prove that medical products are safe and effective. It also explains why some types of clinical trials are better than others.
The gold standard for clinical trials is a randomized double-blind clinical trial. And like she said, the patients are randomly assigned to get drug 1 or drug 2, which could be a placebo or a comparator drug. Patients don't know which drug, and doctors don't know which drug. And this is the gold standard because it reduces bias, because no one knows who's getting which type of drug. Yeah. So then if you go to a single-blind clinical trial, it's similar to a double-blind, except only one set knows who got the drug. So usually the doctor knows that which patients are in which group, but the patient doesn't know whether they got which drug or not. So in this case, you introduce a little bit of bias from the doctor, who would know which patients were in the treatment group and which patients were in a placebo group. So it introduces a little bit of bias because depending on your method of reporting the drug, like if it's pain or if it's something a little more subjective, they can choose how severely to record it based on what their opinion is of the drug. So So then you can also have a randomized controlled clinical trial, and in this case, both the patients and the doctors know which drug you've been assigned to, but you are randomly assigned to get either drug 1 or a placebo or a second drug. There's also a controlled clinical trial where you're not randomly assigned. Your doctor decides which drug is more appropriate to you, and then you study 2 groups of patients that are matched based on age, sex, or diagnosis. And like Diana mentioned earlier, you can also have an uncontrolled clinical trial where you really just have the doctor has assigned a new drug to a patient, and then they study the effect of that drug. There's a lot of different types of controls that can be used in these studies. You can have a new drug compared to a placebo, and in a device study, it'll be compared to a sham device or a device that can be inserted but that's not functional. You can have a new drug compared to an old drug, and this is what's very commonly used when you're trying to treat a disease and you don't want to give patients nothing. If you have a disease like asthma or something where it's not, when you have treatments available, it's not ethical to give somebody no treatment. So you need to compare it to a drug that's already on the market to a new drug. However, FDA, like Diana may have mentioned, your new drug is not required to be any better than the old drug. So you're just looking for whether or not the drug is similar to the drug that's already available on the market. You can also have a new drug compared to historical controls or controls that are not part of the study that you're using. So sometimes they'll use a study that was done years ago of a similar patient population to try and control it to a drug if they don't have the patients or if they don't want to find the patients. So the next part is talking about statistical significance, and that's a word that they use in all of those ads that we saw. This drug is significant. It helps you lose weight significantly. But that doesn't necessarily mean that the drug is actually effective. So if you have a very small sample, your statistical significance could actually be due, you might not have statistical significance because your result might be due to chance. So in this, on the left, you can see that in the control group here, you have 30% effectiveness out of 50 patients in your sample size. And in the drug group, you have 40% effectiveness out of 50 patients. So there's a 10% difference in the group. So it seems like a really big difference. Maybe this drug is working well. However, you only have a 5-patient difference between the 2 groups. So that could just be due to chance or due to something else. One group's exercising, one's not. And when you calculate out, we use a p-value to determine whether or not something is statistically significant. And you're looking for a p-value that's low, so something lower than 0.05. So in this case, because there's only 50 patients in your group, your result is not significant. Oh, yeah, P stands for probability in the p-value. So if you have the same difference between groups in a much larger sample, you can actually end up with a statistically significant result. So in this case, you increase your sample size to 500 patients. In the control group, you still have a 30% efficacy, and the drug group, you have a 40% efficacy. However, it's much more likely that this difference is not due to chance because you have such a large patient population. So here you can see that the p-value, or the probability that it's not due to chance, is very low. So this result is statistically significant. So we just want to remind you that statistically significant doesn't necessarily mean important. It just means that it didn't happen by chance. So your difference might not be 10%, it might be 1% or 2% and still be able to be statistically significant. So some of the important questions to ask about clinical trial design are whether or not your sample sizes are large enough to tell a difference between the treatment and the control group, to tell whether or not it's due to chance or not. Is the study well controlled to reduce bias, such as differences between the treatment and control groups that might have contributed to the result? And is that statistically significant result even meaningful to patients?
The last section explains why it's so important to include all kinds of patients in clinical trials and how to make sure that treatments are tested on patients like you. This helps patients understand the importance of diversity in clinical trials so that each of us can choose the treatment that is most likely to help us and least likely to harm us.
Will this treatment work for me? So when I say, will this treatment work for me, I mean, is the treatment safe and effective for everyone? So in order to figure out if this is the case, we have to look at something which is known as subgroups. So subgroups are actually a subset of people who are pulled out from a larger group of people, such as different races, ethnicity, age, and sex. In order to test to figure out if the treatments are effective and safe for everyone, we have to perform something known as subgroup analysis. And basically, subgroup analysis looks at whether the treatment is safe and effective for in all subsets of people. Because drugs can actually work differently in different subsets of people. They can work differently in women and differently in men and in different races. Okay. So why does that matter to you? Why do we even care about subgroup analysis? Well, one, it improves our understanding of diseases, and when you look at different races, sex, and age. For example, African American women tend to have more aggressive forms of breast cancer, and heart disease actually presents itself differently in women than in men. And the elderly actually tend to metabolize drugs a little differently than the rest of the population. So another reason it matters is because this is the only way we actually know that treatments and medical devices are actually safe for everyone, not just a special group or the majority of the population, but for everyone. And typically the challenge with that is women and people of color, as well as the elderly, are typically underrepresented in clinical trials. And unfortunately, we've been taking the one-size-fits-all approach. If it just works in the majority, they feel it will work in the minority, and that's not the case. And we know this approach does not work. Okay. An example of the one-size-fits-all or the one-dose-fits-all not working is through the drug known as Ambien, which the FDA approved to actually treat insomnia. So millions took the drug, and unfortunately, they were involved in car accidents due to drowsiness from the next day because the drug hadn't cleared the system. So, because of the dosage that they had prescribed, and being they didn't actually look at the drug in men and women, the drug actually stayed in women's bodies a lot longer than men. And they discovered this a little too late. So the women, because it stayed in their bodies much longer, they were more likely to drive impaired the next day. So that resulted in a lot of car accidents and caused a lot of tragedies. And due to that, they had to reduce the dosage for women. They actually had to. So after 20 years after approval, they actually had to halve the dose. So as we can see, one dose doesn't fit all. It doesn't work. It may work for some things, but not for your medications and your medical devices. It's too serious, and it's too much of a risk. Okay? So from this, we learned that one dose does not work for everyone. And the FDA's job is to protect us and make sure that all drugs, treatments, medications, and medical devices are safe and effective for the entire population, not just the majority. So with the case of Ambien, the FDA failed, but hopefully today we can work on making sure they don't fail anymore by being informed patient advocates and letting them know. So what the take-home message from this is that all treatments need to be proven safe and effective for everyone. And in order to do this, we have to study subgroups.
We hope our Train the Trainers webinar has been helpful. Feel free to contact the National Center for Health Research with any questions.